Hello guys, can you hear me? If you can hear me, just let me know in the chat below, please. Okay, perfect. I'm just gonna wait 3 more minutes. We'll start when it's 7:20, just to make sure everyone had a chance to join. Okay? In the meantime, while we wait for the rest to join, if anyone has a specific question before I start, let me know. If not, let's just wait till everyone else joins. And guys, just a small note, because I think some of you are new to my channel. So if you haven't subscribed to my channel yet, please don't forget to subscribe to my channel. Is Yeah, it will be recorded. So when we're done with the video, it's gonna go on my YouTube channel immediately. So you can watch it whenever you want. Don't worry about that. But the reason why I'm doing it live right now, so if anyone has questions, I can immediately answer them. And it's more interactive with everyone watching too. But if you can't watch it right now, that's fine. Watch it whenever you're able to. Okay, 3.2 is really hard. What is 3.2? Let's check. So 3.2 is talking about the factors that affect enzyme activity. Okay, but we'll talk about it also. Don't worry about it. Don't worry about it, Liliana. Best of luck on your exam and hopefully you ace it. Okay, 3.3 is hard because of the words. Okay, so 3.3 is enzymes and biotechnology. Okay, don't worry about it. We'll also explain the words together and hopefully we'll make it as easy as possible. Okay, let's just wait another 2 minutes. If you guys know your friends want to join, just make sure you tell them to join right now before we start so they don't have to miss anything. Can you explain the pH and then theories like those most? Yeah, okay, don't worry about it. Hala will explain everything, and if anything you need more explanation, just let me know, and I'll re-explain it again. Okay? Don't worry about it, guys. The concept is not difficult. I just need you to focus with me, and hopefully we'll try to make it as easy as possible. No problem. You're most welcome. Okay, so I'm gonna start right now. If I think most of you have joined, if again any of your friends still did not join, please let them join as soon as possible because we're going to start right now. So first we're going to be talking about enzymes. So what are enzymes? The first definition we have to know. Enzymes are molecules, okay? What type of molecules are they? They're mostly proteins, okay? And what do they do? So where do enzymes function? They function in chemical reactions that occur in our living cells. So what do we call enzymes? We call them biological catalysts. So look what we're going to do. So enzymes are made up of what? They're made up of proteins. Okay? Now, what is their function? Okay? Function is they assist in chemical reactions. What does that mean? So, for example, in our cell, say, for example, this is a cell, we're having a chemical reaction inside. For example, uh, you don't just any metabolism example. I'm not gonna mention names so you don't so you guys don't get confused. But say we have a reaction that's being done in the cell. For this reaction to work or for it to be faster, we have our cells have enzymes that speed up the reaction. Okay, so enzymes assist in chemical reactions. Since they assist in chemical reactions, what do we call them? We call them biological catalysts. Okay, now this means, what does this mean? What does biological catalyst mean? It means that they do not get used up, so they don't get used up. Okay, and what is their function? Okay, so look what they mean. A catalyst is what is a substance that speeds up a chemical reaction but does not get consumed by the reaction. Okay, so they don't get used up, and their function is what speed up the reaction. So, for example, if I have a reaction A plus B gives me C and D, those are my products, and those are my reactants. Okay, now, for example, this reaction will take, say, 100 hours for it to be completed. Now, if I want to reduce the time, I want to make the reaction go faster, what do I insert? I insert an enzyme, or which I can, another name for an enzyme, I can say biological catalyst. Okay? Then this catalyst speeds up my reaction and it doesn't get used up. So it's not like my enzyme is going to be gone. I can reuse it again. Okay? So it doesn't get used up, it doesn't get consumed in a reaction. That's the first main concept you guys have to know. Yes, A and B are also substrates, correct? So it's reactants, or we can call them substrates. Perfect, well done. So here they're telling us almost all metabolic reactions take place in living cells are catalyzed by enzymes. That's why enzymes are very important in our body. So as we said, they speed up the reaction. So without the enzymes, these reactions would occur very slowly. Like I said here, for example, this reaction could take 100 hours without an enzyme. But once I add an enzyme, that's going to be a faster reaction. For example, it can go to 20 hours, or it can also go for less, maybe 10 hours. Okay, so you see there's a big jump when I insert a catalyst or an enzyme in my reaction, and that's why we have those enzymes or catalysts in our cells, because our cells perform several chemical reactions. Okay, so what did we say? Enzymes are made up of what? Of proteins. Now, what did we learn about proteins? Okay, so what did we learn about proteins? We said proteins have primary structure, they have a secondary structure, they even have a tertiary structure, and also some proteins have a quaternary structure. Correct? So we said in the tertiary structure, what happens? We have folding occurs in the protein. Okay, so that's what they mean right here. So most enzymes are made of large proteins, and like all other proteins, it has a 3D shape that depends on what? On the arrangement of the amino acid subunits along the polypeptide chains. So in order for you guys to know this, think about structure determines function. Okay? So depending on the structure, we can know, so the structure of a thing affects the function it does. The same thing here. So the 3D shape, the shape of it depends on what? On the constituents of it. So what are proteins made up of? Proteins are made up of amino acids. Okay. So the arrangement of these amino acids will determine the 3D shape of the enzyme. Okay. And also, this sentence is very important. Okay. So I want you guys to know this sentence. Structure affects function. And now you will be looking at this sentence even more when we talk about 3.2, where we say the differences in temperature, or the factors in temperature and pH, will affect the structure of the enzyme. That's why the function of the enzyme will be affected also. Okay, but don't worry about it right now. Right now, just make sure you know that enzymes are made of proteins. Okay, so since they're proteins, they have a